This presentation will take a look into the genetics of sporting canines. Long before the days of intense and specialized breeding, sporting dogs made an efficient partner to hunters out in the field. Using their superior instincts and alert personality, these breeds were used to locate, flush, and retrieve quarry. Initially, selection for breeding was based on sociability, responsiveness, and prey drive. However, as man developed more specific tasks for their hunting partners, canine traits and breeds began to expand. Dating back as far as the 16th century, sporting dogs were used widely as hunting assistants on all different types of landscapes. Maintaining an active role outdoors, hunters desired canines with trainable temperaments, high fitness, intelligence, and ability to work well alongside humans. Additionally, terrain type and prey type required specific traits such as water-resistant coats for wetland hunting or the ability to build tunnels and burrows to catch underground prey. Although these breeds are no longer a necessity in the field today, years of domestication have helped researchers to find over 50 genes that can be attributed to the athleticism seen in sporting dogs. A majority of the sporting dogs used were pointers, setters, spaniels, and retrievers, which dominated the sporting dog group, and each grew to prosper in their own task. The main job of pointers was to locate quarry and stand stiffly in front of it, pointing in the direction of the game for the hunter to capture it. Seen in many Brittanies, this pointing behavior can be bred for, but it sometimes requires proper training and dedication to learning the behavior. Setters hold a similar role, especially in bird hunting, but instead of pointing, they were trained to set or crouch in front of the quarry. Popular setter breeds consist of the Irish and English setter, which often have a silky coat and strong olfactory senses to track airborne scents. On the other hand, spaniels are known for their exuded energy and limitless prey drive, which make them valuable flushers in the field. The act of flushing prey refers to first finding the prey and then driving it from its natural habitat for the hunter to shoot. For instance, Springer Spaniels, with their small size and instinct to hunt close to their partners, make them one of the best flushing breeds. Lastly, retrievers do what their name suggests and retrieve dead or injured quarry to their handler. As one of the most common dog breeds used today for service tasks, the role of retrievers required high levels of cooperation with humans in order to assess and control the hunting field. Interestingly enough, golden retrievers were bred as a cross of water spaniels and retrievers to offer hunters a dog that could retrieve on land or in water. The development of various dog breeds and their unique set of skills can evidently be found through hunters' selective breeding and training to assist them in the field. As gun dogs became increasingly essential to the sport of hunting, specific traits were desired by breeders to perfect their hunting job. For retrievers, one trait of high value was a soft jaw so that they wouldn't crush or clench on dead birds. On the contrary, hunters making use of guns in the field yearned for dogs with low sound sensitivity. Regardless of task, hunting dogs needed to be fearless with high stamina as well as a willingness to work. Since the days of hunting for survival and food source, the unique genetic selection of sporting dogs has helped researchers in identifying 59 genes under positive selection that can be attributed to their athleticism. Many of these located genes work in regulating the function of muscular, cardiovascular, and neurological systems of canines. To conduct these findings, researchers from the National Human Genome Research Institute observed whole genome sequence data of 21 sporting dogs, 27 terrier dogs, and 79 village dogs to provide a comparison for the gene and gene variants that appeared in sporting groups. The village dogs also provided a high level of heterogeneity in the population to offer evidence that the groups actually were diverse. To test for athleticism and physical fitness, different breeds performed agility trials while being given direction from their owner. One gene variant of particular interest to sporting dogs was found in the ASIC3 gene, which is related to muscle pain after exercise. Further examination of this gene led researchers to discover that a loss of function rule in the ASIC3 gene, also found in wolf samples, prevented fatigue-enhanced muscle pain. It is believed that through positive selection, this allele had reached near fixation in sporting breeds, as it was shown in 82% of the canines researched. Another gene mutation of interest was found in the CDH23 and MSRB3 genes that are related to sensory impairment. Variants of these genes, also associated with Usher syndrome in humans, were located in only sporting dogs out of all the groups observed. This led researchers to detect that mutations found in these genes are related to accelerated hearing loss after exposure to loud noises such as a gunshot. For sporting dogs specifically, this mutation would be of high significance as it reduces the startle response to gunshots, making for a better hunting partner. One last gene that researchers located related to agility performance was ROBO1. Using the agility titles won by each breed, they found that breeds with a higher frequency of the advantage A allele found at ROBO1, also selected for in sporting dogs, were more likely to win titles. This provided evidence that ROBO1 may affect canine ability to understand environmental information, such as agility trials, so that task-specific responses can be selected for. Overall, the genetic variants identified showed the effects of positive selection on muscle endurance traits and neurological factors that enhance canine athletic ability and success in the hunting field. While sporting dogs have offered valuable traits to hunters for thousands of years, the sheer popularity of these breeds have led to several health and behavioral problems. Careless overbreeding has resulted in increased congenital issues and unstable temperaments in some well-known sporting breeds. To avoid unwanted health issues, responsible breeders should carefully examine all pedigrees of a potential mating to ensure that progeny are free from physical and performance faults. One way to test for performance faults is through field trials, which allow breeders to locate dominant traits and select based on marking ability, trainability, and desire to work. More importantly, breeders should be able to provide evidence of at least the hips and eyes being certified and clear from congenital diseases. Although only few breeders use inbreeding to relatives as a breeding technique for sporting dogs, the suggested method of breeding is line breeding to distant relatives. Line breeding has been proven to be the more efficient method for perpetuating desirable characteristics. However, after about 4 generations, it becomes harder to make large-scale genetic improvements because uniformity of the line has been established. To alter these genes that have become uniform through line breeding, crossbreeding to a different bloodline can help mix the genes. Years of domestication and careful selection have enhanced the necessary traits of sporting dogs to be successful out in the field. Researchers have already located over 50 genes, such as ASIC3 and CDH23, that can be credited to the extreme athleticism seen in pointers, spaniels, and retrievers. These results provide evidence that sporting dogs have been adapted to their occupation by improved endurance and cognitive performance to create breeds with distinct capabilities. Although overbreeding has led to some health issues, responsible breeders should be aware of the preventative steps that can be taken.